Welcome back once again, students. Uh, I'm Teacher Olilo, teacher of biology. Today I want us to look at genetics, Form 4. What is genetics? So from Form 1, under smaller branches or minor branches of biology, we define genetics as a branch of biology that is the study of inheritance and variation. So what we need to know about genetics. The science of genetics tries to explain the differences between organisms and similarities between organisms in the same species. For example, let's look at the member of the same family. So members from the same family may differ in terms of appearance of the face, shape of the nose, ear, skin color, etc. due to a variety of heredity factors that each of them acquires from the parent. Now, before we go into details, what is the meaning of inheritance or what is the meaning of heredity? So that transfer of a trait from the parent to the offspring, that's what we refer to as heredity. When I talk about offspring, offspring is a young one produced by the parent. Like, for example, human beings will produce babies, a cow will produce a calf, a lion will produce a cub. So those are the offsprings. Now, when you talk about a trait or a characteristic, what is a trait or what is a characteristic? Now, those are differences that are seen in organism, those distinct features that are seen in an organism. For example, the color of the flower, height, the skin color, those are the traits. Then number two, what is variation? So variation, these are observable features that are seen among the organisms. So organisms have different observable features. Like maybe organism is tall, someone can roll the tongue, someone has a light skin. So those are variations. Now, normally we have two types of variations. We have one called discontinuous and we have continuous. So now, these discontinuous variations, these are variations or other distinct features in living organisms that have no intermediates. Or we can say that is a type of variation without distinct groups of individuals, or it has no intermediate forms. Good examples of discontinuous variations we have like the sex. So one is either a male or a female. Another example we have ABO blood group. So either you belong to blood group A, or you have blood group B, or you belong to blood group AB, or you belong to blood group O. So you find that you can't be in between, that maybe you have blood group A at the same time you are in blood group B. No, either you are in A or either you are in B. There is no any intermediate. Another example of a discontinuous variation, you have ability to roll the tongue into a U shape. So you find that some people can roll their tongues while others cannot. Then again, we have the presence of long hair in the nose and in the ear pinna. Some people have, either you have a hair or you don't have. Good. Then another example, we have the fingerprint. Is a very good example. In fact, you find that in human beings we have four types of fingerprints. Either it is an arc, is a loop, is a hole, or is a double hole. So among the four, you can only have one. So you find that each individual inherits only one of the four main types of fingerprints. You cannot have both of them. Neither can you have an intermediate. You must have only one. Then you have ability to test a chemical substance. That is, some individuals can test, we call them the tester. Others cannot test, we refer to them as non-tester. So you can't be a tester at the same time you're a non-tester. No, you only belong to one group. In plants, for example, you have pawpaw. So a pawpaw tree is either a male or a female. We don't have an intermediate. Then another one is, before we go to the other type, one thing that we need to know about discontinuous is that discontinuous variations are determined by genetic factors. They cannot be influenced by the environment. They are specifically controlled by genetic factors, that's the genes. Then you have continuous variation. So this type of variation exhibits a wide range of differences from same characteristics from one extreme end to the other end. For example, you have the height. So height within a group of people ranges from the shortest to the tallest within several. Then you have the skin color. So the skin color, again, that the pigmentation, some people have very dark skin, some people have light skin. So again, there we have intermediate. Then you have the body weight. Again, in the body weight ranges. So you have several intermediates. So one thing we need to know about these continuous variations is that they are determined by both genetic factors and environmental factors. So, for example, let's think about the height. You find that let me talk about the skin color. You find that people living in cold areas, they tend to have black skin, while those living in hot areas, they tend to have light skin. And that is, the light skin helps in production of excess heat, while dark skin insulates the body. So you find that that's the environmental factor. So continuous variations are affected by both genetic factors and environmental factors. Then, so we need to know, what are the differences between continuous and discontinuous? What are the differences between continuous and discontinuous? So you're saying that continuous is controlled by many genes, while discontinuous is controlled by one or two major genes. Then continuous can be influenced by the environmental factors, while discontinuous cannot be influenced by environmental factor. Then finally, continuous has got many intermediates, while in discontinuous we don't have any intermediate. Then again, you need to know, what are some of the causes of variation. So we have three major causes of variation. One is gamete formation. So gamete formation occurs into two processes. So one we have during independent assortment, that is during metaphase 1, which was discussed in Form 3. Then we have crossing over, again, which happens in prophase 1 during the formation of bivalent chromosomes. Then it can also caused by fertilization, whereby an offspring inherits some characteristics from the father and some from the mother. So when the two characteristics combines, they'll simply produce a different quality. Then finally we have mutation. So when there is any spontaneous change in the gene makeup, can as well lead to variation. So before we look at how heredity takes place, we need to discuss about the chromosomes. What are the chromosomes? So chromosomes, these are genetic factors or hereditary materials found within the nucleus of a living organism. So remember, we say, if you look at the chromosome is made up of some factors called the genes. Then the genes contain some hereditary factors called the DNA. In full, we call it deoxyribonucleic acid. Now, Um, we need to know that the genetic factors, or rather the genes, are normally occupied by uh, the DNA. So that space occupied by the genes in the chromosomes is normally referred to as the gene locus. Then the gene, as I said earlier, contains some natural chemicals called the DNA molecule. That's the deoxyribonucleic acid. So what is the role of the DNA? DNA, these are the genetic materials. Before we look at the role of DNA, let's look at the composition of the DNA. So DNA is made up of three molecules. We have the phosphate molecule, we have the 5 sugar carbon, or the 5 carbon sugar. Then we have nitrogenous base. Normally we have four types of nitrogenous base. We have the adenine, we have the guanine, and we have the thiamine and the cytosine. So now, what we need to know about are these three nucleotides. The three nucleotides, that's the base, the carbon, and the phosphate. They all combined and they join to form a DNA strand. Normally, if you look at the structure of the DNA is made up of two strands twisted on one another. We refer to it as a double helix strand. So you find that in the double helix strand, or in DNA, adenine always combines with thiamine, while cytosine combines with guanine. So when forming the double helix structure of the DNA, they are equal. Therefore, DNA is like a twisted ladder with nitrogenous base forming the steps or the ring. What is the role of DNA in living organisms? The DNA plays three major roles. One is storage of genetic information in a coded form. So we talk about coded form when the three bases align themselves to form a triplet. We call them the codon. So they store information in a coded form. Then again, the DNA enables transfer of genetic information from the offspring, from the parent to the offspring. Then also translate genetic information into a character of an organism. A DNA performs a very, very important role in protein synthesis. So normally, during replication, as we said, the DNA unzip. When it unzips, you find that a nucleotide is formed in between the two strands. The nucleotide copies the base in the DNA, but you find that during the copying, the thiamine in the DNA is now replaced by a uracil in the form nucleotide. So this nucleotide that is formed at the middle of the DNA strand, we call it an RNA. That is ribonucleic acid. Ribonucleic acid is very important because it is responsible for the transformation of information from the DNA in the nucleus into the ribosomes where proteins are being synthesized. In fact, we call it the messenger RNA. So you find that when the RNA copies the codon in the DNA, so the codon, remember the codon are the triplet bases. So those triplet bases, they determine specific type of amino acid. So once the RNA copies it, then it is transferred to the ribosomes where a specific type of amino acid is manufactured. So again, you need to know the difference between RNA and DNA. So one, we need to know that a DNA has got a double strand, while RNA has a single strand. DNA has thiamine base, RNA has uracil base. Then DNA is found in the nucleus, while RNA is found in both nucleus and the cytoplasm. Now, let's go back to heredity. Let's look at the law of heredity. So law of heredity comes from an experiment which was done by a scientist monk called the Gregory Mendels. And by the Gregory Mendels is considered the father of genetics because he's the one who discovered this concept of heredity. So he carried out various breeding experiments and observed variations in different characteristics of garden pea. So what Mendel did various. So the first experiment, Mendel simply selected a group of dwarf plants and self-pollinated them. So during mature pollen grain onto the stigma of the same plant, he then collected the resulting seeds and planted them. Then he noted that these seeds germinated and grew into dwarf plants only. He repeated the experiment for several generations and observed the same result. This shows that dwarf pea plant could only produce only their own type. They were pure breeds. A pure breed is an individual that is breeding true for a particular characteristic, are always homozygous at all gene loci. He then selected tall plants and self-pollinated them. He then planted the resulting seeds and observed that they grew into a mixture of tall and dwarf plants. He took seeds from the tall offspring only and repeated the experiment for many generations until he obtained tall plant only. He then cross-pollinated the pure breed tall garden pea plants with the pure breed dwarf variety. He planted the resulting seed and observed that the offspring were all tall. From here, he noted that the tall plant are hybrid. Hybrid is an offspring of a parent. A hybrid is an offspring of parents differing in one or more traits, or simply a heterozygous individual. He crossed two of these tall breeds offspring and planted the resulting seeds. He observed that this second generation consists of a mixture of tall and dwarf plants. He counted the plants and noted that the ratio of tall to dwarf plant was approximately 3 to 1, respectively. From the experiment, Mendel concluded that characteristics of an organism are determined by hereditary factors. These are the genes, which occur in pairs. Only one, only one of the pair of such factor can be carried by a single parent or gamete. This is known as Mendel's first law of segregation. The second law is known as the law of independent assortment. Reasons for Mendel's success. He used favorable material, garden pea, which is self-fertilized. His study focused on particular traits. He studied a single characteristic at a time. He kept accurate data all of his experiments. The pea plant he used has several observable contrast characteristics or traits. Then you have monohybrid inheritance. In this experiment, Mendel postulated that the inheritance of a characteristic like height in garden pea plant is controlled by a single pair of heredity factor contributed by both parents. This type of inheritance is known as monohybrid inheritance. It involves the transfer of just one pair of contrasting characteristics, e.g. tallness and dwarfness for height. A single pair of hereditary factors known as gene controls such traits. Genes occupy in pair of chromosomes and such genes pair are known as alleles. Allele are alternative form of a gene that control inheritance of contrasting characteristics. For example, in height, T controls tallness, T dwarf, and TT are thus alleles. The genetic constitution of an organism is known as genotype, while the outward appearance of an organism is known as the phenotype. Genotype determines phenotype, or genotype of gene genotype gene of gene type of genes of an individual. Phenotype, the physical expression of a gene.